Alright, part 3 of chapter 11 here, looking at the different effector functions of the second line of defense. Alright, so after microbes have gotten into the body past the first line of defense, then the second line of defense gets activated, and this is primarily going to be due to the release of different molecules in a particular environment in a localized area to begin with. And those molecules have a range of function, but overall they're there to recruit other leukocytes to the site of infection, bring more, basically more weapons to the fight, restrict pathogen growth so that any microbes in the area, they can't expand nearly as readily, trigger fever, and typically when you think about fever, it's going to be systemic fever, alright, but sometimes that can be relatively localized and that process be seen with inflammation. So, in this process of second line of defense, alright, it's going to initially involve granulocytes, the neutrophils, eosinophils, basophils, and mast cells. These granulocytes can release material, some of those chemical mediators, but they will also go through the process of phagocytosis. And phagocytosis is important to clear out foreign material. The major phagocytes mentioned before, the macrophages, dendritic cells, and neutrophils. This is one of your specific chapter objectives. And that the phagocytes are going to target, sorry, I can't find the cursor here, target bacterial cells, the viral pathogens, general debris, the garbage in your system that needs to be cleared out. Alright, part of the chemical response of the innate immune system will help degrade bacteria to create some of that garbage and to dampen the strength of a bacteria cell, which would then make phagocytosis a little more effective, but also enzymes of the lysosome. Alright, the lysosomes that are found inside the phagocytes will destroy the targeted material once the granulocyte, the phagocyte, pulls the material into the phagosome, and then it creates the phagolysosome. Alright, in some cases, microbes have virulence factors that help them to avoid phagocytosis, and so they can go unchecked and replicate at higher paces. Alright, here's an example of a phagocyte here. Alright, you've got the fluff kind of the membrane of the phagocyte that will create those pseudopods, alright, to help engulf the bacteria cells here, and it's going to completely encapsulate the microbes here and draw it into the host cell, or the phagocyte here, in this phagosome here. So it kind of looks like this is just a pouch, but as it pulls it into the interior of the cell, it actually creates a bag, the phagosome. Alright, now some of those defense molecules that work in the second line of defense are cytokines. The cytokines are going to stimulate inflammation, generate fever, recruit leukocytes to fight infection. Alright, so that's what we saw previously. And it will also stimulate tissue and blood vessel repair because there is a reason for those cytokines to be introduced or to be produced and released into the environment. And that could be bacterial infection, it could be trauma. And so then in trauma or an injury, you need tissue repair, blood vessel repair. Cytokines will promote leukocyte and lymphatic tissue development. This is good for additional fight for fighting that infection. And there are interferons that are antiviral. Interferons are molecules that can stimulate neighboring cells to say, hey, there's a virus in the area, and basically to put up protective markers to help to avoid infection by the neighboring viruses. Right, iron-binding proteins. I don't know if we mentioned this before or not, but iron-binding proteins can limit the availability of free iron, and so that reduces bacterial growth. But you don't need to worry about that. Complement, we'll look at a couple of slides on complement, but complement is a series of serum proteins, and they're going to function to stimulate inflammation, all right, this process up here, and then tag targets for elimination. This is called opsonization, but this will cause phagocytosis to be much, much more effective, more efficient, and able to clear out an infection so much faster than without the complement. And then cytolysis, direct killing of targeted cells. Cells that have the protein, the complement proteins put on the cell surface, that basically calls the immune system to say, hey, this is a bad cell, it needs to die. And different parts of the immune system will allow for cell death. Right here is a table looking at the different examples of cytokines. Cytokines are just a group of cellular signals that are produced, and cytokines can be categorized as chemokines, interleukins, interferons, and then TNF. And you don't need to worry about what all of these are, but rather understand that individual molecules here are going to have different results, different processes in the body that they instigate, and that they are also made by different types of cells. Alright, so you've got macrophages that are producing some of these. You have natural killer cells, different T cells, which we'll talk about in chapter 12. These are all coming from different types of cells, still in that process of trying to clear out an infection. Alright, here is looking specifically at interferons, the cytokines that are produced in response to virus-infected cells. And these molecules act as alarms that stimulate nearby uninfected cells to mount an antiviral defense without even being infected. And it's pretty involved, it's pretty interesting. And what it does is, this is the infected cell. This cell produces interferons as a response to this viral infection. The interferon is released, and it stimulates uninfected neighboring cells to decrease protein synthesis. If that happens, then once the virus got in, then protein synthesis wouldn't be going on, and so the virus wouldn't be able to reproduce, to allow it to be copies made of itself. It will trigger apoptosis of infected neighboring cells here, and interferon also activates additional leukocytes. Those white blood cells that will come in can come in and help clear the damage that is being produced, such as in this scenario here. Alright, so what is complement? It is a set of serum proteins, alright, up to 30 different proteins. They are mostly made by the liver. They circulate in the blood in an inactive form, and once they get activated, they're generally cleaved or cut apart, and so then they can be activated. And they work in a cascade fashion, meaning one protein will act on the next protein, which will act on the next protein, kind of like a domino effect. And this cascade of events acts as a booster for the rest of the immune defenses. So there are 3 different complement pathways, and they all have the same outcomes. The first is opsonization. All right, part of a couple of the different complement proteins will become attached or tag invader. All right, so a bacteria cell. And once it has that particular tag on it, then the phagocytes come in and very easily phagocytize it and clear it. There's the formation of the membrane attack complex, sometimes referred to as the MAC. That creates holes in the cell wall of the bacteria, and that causes that cell to die. Right? Cell here with cytolysis, making holes in it, breaking apart, you break the cell apart. And then also complement will induce inflammation. And again, remember, inflammation can be a good thing, can help the clearance of bacteria. And so this is just a figure looking at the 3 different pathways. I don't want you to memorize this unless you want to. But here you've got some of the different complement proteins. You have C3, and then the C3 molecule gets cleaved into a C3b and a C3a molecule. Both of these have different functions. You see here C3b is actually going to be a tag for bacteria to allow for phagocytosis to happen much more effectively. C3a will actually go into the blood to cause inflammation, and that moves out into other areas of the tissue. C3b will also interact with C5 to cleave C5 into C5b and C5a. C5a is also a molecule that will induce inflammation. C5b goes to a bacteria cell here and will help to facilitate other complement proteins to come in and create this hole. All right, so you see the hole here has quite a few of these proteins that come together, different numbers of the C9 molecule that actually makes the hole. And the different pathways here indicate how this pathway of proteins is activated. Right, there's the classical pathway, which involves antibodies. We'll talk about antibodies in Chapter 12. There's the alternative pathway, which is this tagging process. And then there is the lectin pathway. And this is a different set of proteins initially that will bind to mannose binding lectin, and that helps to bind to a particular type of pathogen, bacteria cells, and then that moves into this process. And you see all of these processes, all of these 3 pathways move into or move through the C3 complement protein and the same end events: inflammation, cytolysis, and opsonization. Alright, and that is where we are going to stop part 3 at the end of complement.